हेलो गाइस, आज हम स्टार्ट करते हैं हमारा पहला वीडियो लेक्चर ऑफ दिस चैनल पेंडोरा फार्मा. सो आवर फर्स्ट टॉपिक is conservation of medicinal plant. We will go with a review that Ayurveda, traditional system of medicine recognized by WHO, increasing global demand for medicinal plants, need for conservation and sustainable utilization. So, in the introduction, we will go with that Ayurveda has been practiced in India for centuries. WHO estimates that 80% of the world population depends on traditional medicines. India possesses around 8,000 medicinal plant species. Medicinal plants form the backbone of primary healthcare in India. Next we will see that what is the importance of medicinal plants. So used in Ayurveda, Siddha, Unani and folk medicines. Economically important for developing countries. Safe, effective and natural treatment options suitable for all age around group with minimal side effects. Threats to medicinal plants diversity and need for conservation. So what are the threats? So over exploitation due to high demand, this is something, and the next is deforestation and habitat destruction. Natural calamities like flood, droughts, climate change, these all. Man-made activities and urbanization, loss of genetic diversity and extinction risk. Need for conservation, what are they? Present or to prevent depletion of wild medicinal plant, maintain biodiversity and ecosystem balance, ensure sustainable supply for future generation, support traditional healthcare system, reduce dependency on wild collection. Next, we will move forward and see that what are the type of conservation. Two main approaches are in situ conservation, which is on site, and ex situ conservation, which is off site conservation. So in situ conservation, what are they? Conservation of plant in their natural habitat, maintain natural evaluation or evolution and adaptation, conservation of entire ecosystem along with the species. And what are the example which are present in India? So national parks, wildlife sanctuaries, biosphere reserves, wildlife nurseries, all these are. Next we will look at all the example one by one, like natural parks and wildlife sanctuaries. So these are protected areas established by government, prevent illegal collection and habitat destruction. For example, Corbett National Park, Uttarakhand, Ranthambore National Park in Rajasthan, Vilavdar National Park in Gujarat. Next, we will move for biosphere reserves, so large protected areas for biodiversity conservation. Preserve endemic and endangered species of medicinal plant, and India has 18 biosphere reserves. Example, Nilgiri, Sundarbans, Nanda Devi and Agastya Malya. Wild nurseries, in situ conservation techniques, protects threatened and endemic species, helps in regeneration and natural propagation. For example, Periyar Wildlife Sanctuary, Bandipur Wildlife Sanctuary. So what is conservation? What is conservation? So it is plant management and protection of animal resources, sustainable utilization without harming the ecosystem, which includes the collection, propagation, storage, disease estimation and distribution. So here we have our next topic, which is ex situ conservation. We will look further deep in this topic. What are they? They are conservation of plants outside natural habitat. Okay, so used when in situ conservation is not sufficient. Okay, reduces pressure on wild population. It reduces it. What it includes? It includes botanical gardens, seed banks, filled gene banks, tissue culture, and cryopreservation. We will look further deep into cryopreservation. Okay, so next is botanical gardens. What are they? They maintain the collection of medicinal plants. They support research, education, and conservation. What are the examples? Examples are government botanical gardens in Tamil Nadu, Empress Garden, Maharashtra, Garden of Medicinal Plants, West Bengal. Next, we will see that what are seed banks. This is new word for you. Let's see this. It stores seeds under controlled condition. It stores the seeds under controlled condition. We preserve the genetic diversity of it. Quick access for research and reintroduction. Okay, and what are the challenges for making the seed banks? Nothing but seed viability, reintroduction into wild habitats. Okay, so these are the things which is new. Next is filled gene banks. What are they? So conservation using biotechnological tools. Okay, so biotechnological tools we are using for conserving. Okay, what? The seeds, the filled banks. Okay, suitable for vegetative propagation of plant, and these things include the techniques of in vitro conservation in the test tube and all. Molecular marker technology is also included in this. Next, we are seeing for germplasm conservation. Conservation of plant genetic material stored at seed, tissue or plantlets, reserve less space and maintenance. What are the advantages? Reduces disease and pest attack. Maintains thousands of genotypes. What are cryopreservation? Hey, this is the important topic. So, storage of biological materials at 196 degrees Celsius. Okay, using liquid nitrogen, we are using it. Okay, here maintain long-term viability. And what are the advantages? This is vitrification, encapsulation, dehydration, droplet freezing. So next we will go for this diagram. How beautifully it explains that the source tissue, which is suspension cell, somatic cell or a zygotic embryo, it goes through dehydration. High osmotic pressure. Okay, with high osmotic pressure it goes to dehydration, and what it goes? Pre-growth, and then cryopreservation with the presence of liquid nitrogen, and the next arrow goes for the storage and the temperature maintenance of 45 degrees Celsius, increase osmotic pressure. Okay, now thawing. What is thawing? We will look in the further videos. And the next is viability test, regrowth, and then regeneration, and the test for somaclonal viabilities, and we get the plant. How beautifully it happens now. Yeah, so these are the topics. Next we will see that how tissue culture techniques, and what are they? So in vitro culture of plant cells or tissues, these are they. Controlled sterile condition applications. What are they? Conservation of endangered plant, rapid multiplication. What are happening? Rapid multiplication, and preservation of high yield medicinal varieties. Okay, thank you so much for giving your attention and being in my newly made video of this channel Pandora. pharmacy.